Hey y'all, what's up? Coach Todd coming at you with a video on proteins. Got a little bit of the sweet nectar of the gods here, some Mountain Dew. Come here. Tell him. Rowan, show me your muscles. Oh, wow, and them muscles. What makes your muscles so big? Protein? You say protein?
I eat chicken.
You eat chicken? And chicken has what in it? Protein. Say protein.
Protein.
That's right. That's what today's video is about. Let's see here. Protein. So like Rowan says, you can get protein from chicken, which are animal products. You can get protein from fish. You can get protein from plants, right? Certain plants, not all plants, right? Protein's out there in all kind of different sources. And it's important for us to grow, to be big and strong, right? And healthy. So a protein is a polymer. It's a macromolecule, just like everything else we talked about this unit, okay? You know, carbohydrates is a macromolecule. Lipids is a macromolecule. Now we're on proteins, which is also a macromolecule. We know these things are built of base units, right? We call monomers. And the monomer of protein is an amino acid. An amino acid. So if we chain together amino acids, then you have protein, right? So amino acid kind of base, we start out, something like this. Right, something about an amino acid. Right, they all have an amino group. They have an amino group, and they have a carboxyl group. The amino group is like that. Color. Just gotta grab another color here. Carboxyl group. Make sure I'm not wrong. I'm wrong already. Something like that, right? And this is a carboxyl group. Now, something about this. This part of the amino acid is the same, right? For all amino acids, essentially, right? Pretty much the same. The difference between these amino acids comes here in the R group. Now, if you look at different examples of different amino acids, you'll see that you should be able to point out this structure. See that all this is the same. When you start looking at the side chains, R group, you start to see some different things happen. This is what gives each amino acid its properties, whether it's polar, whether it's nonpolar, whether it has a positive charge, negative charge, no charge, things like that. Sorry, I didn't know my computer was playing something. But that's our amino acids. So if we start to bond these together, we start to make chains of these, right? So we'll kind of make it a little easier. Right? So we'll have, common ending of proteins is -ene, right? Thing. So what? Sorry about that. I had a brain fart for a minute. It's been a long day. Anyway, so have an amino acid. Right? We bond it to another amino acid. What? This bond that we use to bond the two amino acids together is AA amino acid, not Alcoholics Anonymous amino acid. Right? We join two amino acids with a peptide bond. Okay? So every time we join an amino acid, we get a peptide bond. And after we get a couple peptide bonds going, we turn amino acids into a peptide. A chain of amino acids is a peptide. Once that chain grows, right, we have a polypeptide, right? Polypeptide. And once we get a polypeptide, right? So once we get a long chain of amino acids, right, we get a polypeptide. Right? We have taken a bunch of amino acids. So we'll say the blue is a bunch of amino acids. We bonded them together with peptide bonds. We now have polypeptide. This is our primary structure for our protein. This is where the protein folding kind of starts, our primary structure. So now we're going to get, as this continues to develop into a protein, now we're going to start to get some spiral shape. Thank you. Okay. So we start to get this polypeptide is going to start to kind of take shape. It's going to form an alpha helix, or kind of like a mated pleated sheet. This would be our secondary structure, and then it will start to kind of come together at the R groups with some different interactions. It's really going to start winding up and winding up. Then we'd have our tertiary structure. And if we combine two or more of these tertiary structures together, now we have a big jumbled globular mess or fibrous mess that is called protein or quaternary structure. If you want more information on that, check out the video on protein folding I did, very detailed with our pipe cleaner demonstration. But that's kind of where we're at right now. So once we have proteins, they can do all kind of different functions, right? We can use them for structure. Uh, I dare not write this because I will probably get it wrong, spell it wrong. But keratin is the structure that our hair, the protein structure for our hair. Um, if you think of eggs, the egg white is protein storage for the developing embryo in the yolk. Milks have casein in them. That's the protein for their young. We got transport proteins. We got hemoglobin proteins that hold oxygen. Just all kind of different functions for proteins. I mean, it's endless, really. Um, so up next, I guess, is the word denature. So something that can happen with our proteins, right? If we have our big, so Rowan started to draw us one right here. We have our big globular protein, or we can have some kind of fibrous looking protein, right? If it is stressed, if it is put under kind of some conditions that it's not designed to withhold, so high temperatures like a fever, or changes in pH, or changes in salinity, any kind of environmental conditions that change too much or too drastically can denature the protein. Right? So denature. What this does is, you know, when we talked about the protein folding up into this big crazy structure, denaturing means it's going to kind of do the opposite. It's going to break these interactions that hold it together. It's going to break down these alpha plated sheets. It's going to turn it back, right, into our polypeptide. Now, this can be reversible if conditions are favorable. It can also be irreversible. As we learned in our lab, um, once so much has been denatured, it just won't work as well. The process that was happening with the proteins won't go as well. So that is denaturing. Kind of the last thing we'll talk about this a lot with proteins, is they are enzymes and hormones. We don't talk about them much being hormones, but some of our hormones are proteins, like insulin's one. But anyway, going into enzymes. So enzymes make reactions happen faster. So if we were to think about a graph where we have energy going up like so, and kind of start with our reactants. Okay, reactants react together to form the products, right? A plus B is the reactants, C is our products. So in order for our reaction to occur, we have to input energy. So if we input this energy, that's our input energy, that hump. Then, okay, so kind of this kind of represents, right, our activation energy. It's the amount of energy we put in to make it happen. Then gotta get this drop for our products. This part here, right, represents the energy released from the reaction. So some reactions give off heat, or maybe they get colder, or whatever. They absorb energy, which will look a little different. But anyway, this is kind of natural, right? Now, what we can do is we can introduce an enzyme. If we introduce an enzyme, it's not going to change the reactions. It's not going to change the products. It's not going to bond with them. It's not going to do anything with them. It's just going to be just like it is right here. What this enzyme is going to do is going to make it happen faster. And the way it does that is if we insert our enzyme, right, the graph's going to look the same until we get here. Now our activation energy is lower, right? We have lower activation energy with an enzyme. Everything else is still the same, right? We're still doing the same products, start with the same reactants. The use of an enzyme lowers that activation energy. Now, the way an enzyme works. Thank you, babe. Yep. I don't really know about. I can't really remember what Pac-Man looks like. But you can kind of think of it as like a Pac-Man type shape thing or whatever. But what an enzyme, so if this is our enzyme, it's going to do, right? It's going to bond to our substrate. Huh? So our enzyme is going to bond. So this is our enzyme. This is our substrate. It's going to go grab this substrate, right? And it's going to hook it to whatever's bonding, right? A plus B is going to take A and it's going to hook it to B, right? It's going to hook it to the other piece, right? So it's going to kind of go grab this, bond it with this, right? Make it happen faster. It's going to give us, then it's going to release this, right? So now it has released our new product, which looks something like that, right? So what we have our enzyme, we have our substrate. That's spelled wrong. Our enzyme is going to grab our substrate. Only can work with one thing, based off its shape, right? It's out of the triangle, it's going to grab this one. It's not going to grab that, right? So it's going to bond to our substrate. It's going to bond them. It's going to make it happen a lot quicker, a lot easier, right? And then it's going to let it go. And it's going to go back. It's going to redo the whole thing. It's kind of called the lock and key method, right? Because think about a lock and key. Now, there's exceptions to every rule, right? But a key essentially only works with one lock, right? So you can use this key with this lock over and over and over and over again, right? So the enzyme's kind of like the lock. The key is kind of like the substrate. You're going to put the substrate into the enzyme. It'll work. It'll do its thing, lock the door, then it releases it. Release. I guess kind of last thing about these enzymes is they always end in ASC. So, threw this thing together for y'all for proteins. I feel like it's a jumbled mess. Like I just went in a bunch of different directions. I do not have time to redo it though. So, you get what you get. Forgive me if it's confusing, all right? But just kind of re-illustrating everything. Proteins are made of amino acids, right? Amino acids are the monomers for proteins, right? They build the proteins. Once we start bonding amino acids together, we get a polypeptide. We get this primary structure of building a protein. Then it's going to go through to the secondary, the tertiary, and the quaternary structure of our protein. and that's when we get our big globular or fibrous protein is at the quaternary stage. There's a video that breaks that down pretty good I made. Check the vlog section. So now we have our protein. It can do a lot of different functions. Those functions include, hair, structure in the cell, cytoskeletons, protein storage. All kind of stuff. Transport, motor, movement, defense. Protein does a lot of different functions because it has tens of thousands of different structures, and we know structure determines function. One thing that happens to proteins, if the conditions change too much, too drastically, it will cause the protein to denature, which means that it would fall apart basically from its quaternary structure down to a tertiary or secondary structure in which it will no longer be biologically active. It's not going to work anymore. It can be reversible. It can be permanent. It just depends on the protein, depends on the environment, depends on a couple different factors. Proteins are hormones, such as insulin. Insulin tells your body to metabolize blood sugar or not or whatever, right? So hormones can be proteins or lipids. Remember we talked about some hormones being lipids and steroids, cholesterol type stuff. And then finally we ended up with proteins being enzymes. Enzymes in an ASC, catalase. We did our lab with catalase. So that is an enzyme. And enzymes do not really change anything in the reaction. They just lower the activation energy so that the reaction can occur faster. They help out the reaction, if you would. Um, they don't change the reactions. They don't change the products. So that's pretty much it. The first 15 minutes of the video was a mess. I feel like the summary was pretty good anyway. So let me know if this made sense to you. If I need to start over one day after school. All right, catch y'all next time. Nucleic acids is next.